So it's our great pleasure and honor to have Professor Soumya Raychaudhuri. He's a physician, scientist, and also internationally recognized leader in human genetics, as well as immunology and data science. So Dr. Raychaudhuri obtained MD, PhD from Stanford University Medical School, followed by his specialization in rheumatology at Brigham and Women's Hospital. At the same time, completing his postdoc training at the Broad Institute. So in 2010, he became a faculty at Harvard Medical School and currently is a professor of medicine and biomedical informatics at the Harvard Medical School and director of the Center for Data Sciences at Brigham and Women's Hospital and the institute member of the Broad Institute. So as we are all aware, Dr. Raychaudhuri's research has revolutionized our understanding of immune-mediated disease by employing large-scale human genetic studies with single-cell genomics, and also helped to unravel many molecular and cellular mechanisms underlying the conditions in rheumatoid arthritis, type 1 diabetes, and tuberculosis. Dr. Raychaudhuri also have a collection of achievements and recognized through numerous awards and membership, including the Doris Duke Award and the American College Rheumatology and Henry Kunkel Young Investigator Award. He's also an elected member of American Society of Clinical Investigation, also Clarivet highly cited researcher, as fellow of American Association of Advancement of Science. So with that, please join me in welcoming Dr. Soumya Raychaudhuri.
Okay, thanks so much for inviting me to come and speak here. It's a great pleasure to be here and tell you about some of the work we've been doing in defining cell state-specific molecular effects of disease risk alleles. Great, this works all right. Okay, so in the first part of the talk, I'm just going to talk about autoimmunity and broadly, and give you my perspective on autoimmunity and how we're using human genetics to really try to understand it. And then I'm going to move on to talk about some strategies we've been using to try to find causal alleles for these different loci that drive complex diseases, specifically autoimmune diseases. In the third part, I'm going to present some really new and exciting work on functionally confirming the effects of causal alleles with genome editing. And then finally, I'm going to move on to talk about actually finding SNPs whose effects on molecular traits are cell state specific or dynamic across cell states. So let me start by talking a little bit about autoimmune diseases. So as all of you probably know, autoimmune diseases are a set of diseases where your immune system aberrantly recognizes presents self-antigens to the T cell, resulting in an attack on your own organs. Some of these diseases are very specific. For example, thyroiditis affects the thyroid, but others are more sort of promiscuous in terms of what they do. So systemic lupus erythematosus can affect a wide range of different organs. These diseases are individually rare. So if you consider a condition like systemic lupus erythematosus, it affects about 1 in 1,000 people. Type 1 diabetes in the US affects about 3 in 1,000 people. Rheumatoid arthritis, which I think the most about, is about 1% of the population. But in aggregate, they're fairly common, affecting about 1 in 20 individuals when you consider all of the full spectrum of autoimmunity. Rheumatoid arthritis, the condition I think the most about, is actually a disease that was described in the year 1800, long before single-cell data was available. It was the year 8 of the French Republic, and actually done by a medical student in their dissertation. At the time, the student recognized this condition as asthenic gout. So that is like something that's like a little different from regular gout in that it predominantly affected women. It had a chronic course. It was polyarticular. And so that this was the first real recognition that this was a different disease than the much more prevalent gout. And this is Pierre Auguste Renoir, and you can see he actually had rheumatoid arthritis, and so what you can see if you do a blow up of his hand here, you can see that his hand is quite damaged by rheumatoid arthritis. But despite the damage, he was able to do paintings like the one on the left. That's a self-portrait. And most of those paintings in this stage of his life were done with a brush strapped to his wrist. Autoimmune diseases many autoimmune diseases, though not all, are increasing in prevalence in general. This is data on type 1 diabetes going back to the 1960s, and you can see across a range of countries the number of people who are being diagnosed with type 1 diabetes is dramatically increasing. One very promising thing is over the last, say, 20 years, we've gotten much better at treating autoimmunity, and that's largely been driven by a better understanding of immunology and the development of targeted therapeutics. And I've got a few examples here. So, for example, cytokine blocking has become a very popular therapeutic strategy. This is what we use with anti-IL-6, such as tocilizumab, or anti-TNF drugs, which are widely used for a lot of diseases. Cytokine receptor blocking, also similar kind of strategy. There's also a lot of approaches using to block T-cell signaling, such as CTLA-4 Ig. Depleting B cells or deleting B cells with rituximab or ocrelizumab has also been a strategy. And finally, blocking signaling in immune cells, for example, with tofacitinib, which blocks JAK inhibition. And this is a really big therapeutic market, but there continues to be a demand. In particular, despite these advances, we're still really not able to cure autoimmunity. So there's still a need to have a better understanding of these conditions and to come up with treatments that lead to durable remission or to cure. Our approach has been really to think about genetics and really trying to get at this idea of figuring out what the genetic mechanisms of disease are. So human genetic studies have helped us link many SNP loci to autoimmune diseases, and of course to complex diseases in general. And we think of genetics, again, maybe more broadly, as a great source of causal human biology that could be exploited for many purposes, including therapeutic development. But across all complex diseases, the barrier, I think, in the field of human genetic studies continues to be linking those common variants to causal disease mechanisms. And as many of you know, we have found thousands of SNPs through association studies that have been described for a wide range of diseases, but very few have been linked to very specific mechanisms. This is a locus that I was studying about 10 or 15 years ago called complement factor H and age-related macular degeneration. It was about 4 or 5 years before we wrote this paper. It was one of the first GWAS loci that came out. What you can see is the problem. It was both an exciting result to see all of these SNPs associated with this locus. You can see that they're highly statistically significant, but you can see that there's about 12 or 14 of them. So it's really hard to know which of these is actually driving the phenotype. And of course, they're all perfectly correlated, as indicated by the red block there below the locus plot. Rheumatoid arthritis has been very successful in the sense that we have discovered many of these loci. We're now up to about 150 loci. The latest study that we published in 2022 with collaborators from around the world, led by myself and Yukinori Okada, and Kazuyushi Shigaki, Saori Sakawi, and Chikashi Terao. This particular study represents 5 different ancestry populations, and again points to a whole bunch of interesting loci harboring interesting genes. And one of the major themes that have come out of studies like this for RA and for other autoimmune diseases is that T-cell biology is really important. So about 10% of the alleles are coding alleles. So those are maybe a little bit easier to make sense of. But if you look at the 90% of alleles that are non-coding alleles, they're disproportionately close to T cell genes. They're disproportionately present in regulatory regions that modulate T cell genes or are T cell specific. So all of that points to T cell biology as a really central piece of autoimmunity. And the HLA locus, of course, is really important for all autoimmune diseases. And that, of course, is intrinsic to presentation of antigens to the T cell receptor. So on the one hand, we have all of these loci. We have some clues that T cell biology is relevant. What are the barriers to getting to mechanism? The first, as I've already alluded to, is fine mapping the loci to find the causal alleles. This is challenging because you often have a lot of SNPs that are in linkage disequilibrium with each other. The second piece is defining the function of those causal alleles. So even if you think you have the allele, we really need to understand its effects on gene regulation. And we want to functionally confirm those effects as well, which again has become a real bottleneck relative to the rate at which we're discovering things. And I would argue that we need to do, and I would say this is if you get a bunch of geneticists in a room, I think this would be a little bit contested, but I think we need to do better than just being able to say this is the target gene. We actually want to understand the functional context and the upstream mechanism, because that really gives us an opportunity. And I think there's a real role for the Human Cell Atlas in thinking about that functional context. And just to give a sort of provocative example, here I'm looking at data that we published recently looking at T cells from inflammatory tissue. And you see a lot of interesting T cell states that are really very tissue specific. And if you look at IL-6R, which is a gene that's come out of our study of therapeutic importance, if you look at IL-6R expression, you can see that they're expressed in T cells, but not in all T cells. It's really expressed in a specific subset of T cells, and that subset of T cells is this tissue-specific population of T peripheral helper and T follicular helper cells. So there's a really important context to where this gene is acting. And when you look forward, when we think about understanding genetic mechanisms, if we understand the functional context, there's a real opportunity. If we can define the context-specific molecular effects, we can think about a therapeutic targeting strategy where we're not just targeting a protein non-specifically like we do now, but actually trying to target the protein in a pathogenic context. That is, for example, rather than targeting all cytokine receptors, it might be better to target the cytokine receptors that are expressed in the specific immune cells that are causing the specific problems. And that might lead to a potential for an effective, durable treatment that's also very narrow and therefore safe in terms of immunosuppression. So just to summarize this first section, a cure to autoimmunity remains elusive despite all the advances that have been made. Genetics has been a powerful way to start thinking about how we might make progress towards these cures. Autoimmune diseases have been successfully investigated with genetic studies, but the major barrier in human genetics is to develop strategies to link loci to disease mechanisms. Rheumatoid arthritis and most immune-mediated diseases really point to T cell regulatory elements, and translating these alleles and loci to specific mechanisms is essential, and it really demands that we understand non-coding genetic variation. And atlases like the ones we're talking about here have a really important role in defining the functional context of disease genes. Okay, so now I'm going to talk about not just discovering loci, but how do you actually find the causal allele in a given risk locus. So localizing causal genetic variation is a really important activity for our group. One of the first tools that we all use is statistical fine mapping using methods like fine map. But what you usually end up is with multiple causal variants. There are rare instances where you find a single causal variant, and I'll talk about some of those examples. But being able to look at a set of potentially causal variants and being able to assign function to one of them is very useful. And obviously, coding variation is the most obvious way to think about function. So sometimes fine mapping can really work well, and this is a very unique example in autoimmunity, in RA specifically. So this was the DNase 1L3 locus. It was something we first discovered an association to in 2011, and as our data got better, what we realized is there's really a single variant that explains all of the signal. And it's this arginine to cysteine change. It's a coding change and has a very high posterior probability. It sits right in the middle of this DNase 1L3 gene. DNase 1L3 is a DNA, so it removes sort of pro-inflammatory DNA from the milieu. And loss of function mutations causes an increased risk of rheumatoid arthritis. So sometimes there are these instances where you can find map something, you can make sense of the coding variant, but more often than not, we're stuck thinking about gene regulatory variants and non-coding alleles. In that instance, the best path forward is to really think about how that genetic variant might be associated with gene expression. And this, of course, leads us to thinking about EQTL studies, which I know the HCA is very invested in now. And in some instances, EQTL signals and disease signals overlap perfectly. And that would sort of suggest, though not prove, that a SNP disease allele might be working by modulating the expression of a gene. And the caveat is that a lot of times when we do get this sort of co-localization, you still might not know the causal allele. You might simply be able to say that the signal is shared between the expression phenotype and the disease phenotype. So this is an example of an RA risk locus that we started thinking about a little while ago. And as you can see on the right, as you go from GG to CC genotype, the expression of this BLK gene goes down. And this same SNP is associated with RA susceptibility. And in a lot of instances, what we're imagining is that the SNP is sitting in a regulatory element, such as an enhancer or a promoter, and disrupting the way in which a transcription factor or some other regulatory protein is binding that regulatory element. Now, this is an example where we do see co-localization. So at the top, I've plotted all of the SNPs in this locus and their association with BLK expression. And in red, I've identified the SNP, which is the RA risk allele, the lead there. And then if you condition on that SNP, you can see that all of the association signal for gene expression goes away. So that same SNP that explains all of the disease signal also explains all of the expression signal. Again, which would sort of suggest to you that the SNP or one of those SNPs modulates gene expression of BLK, and that's actually what modulates the expression, what modulates risk of disease. So that's one way to get at some of the causal biology. But obviously, even in this example, you can see that there's a whole bunch of SNPs that are all in tight linkage with each other. It's really hard to know which one is driving the signal. So one of the strategies we've taken, because we really want to know what that causal allele is trying to prioritize those alleles, specifically by looking at which allele might be sitting in a regulatory element, which might be sitting in an enhancer, and this requires us really to build these enhancer gene maps. So alleles within active regulatory regions are more likely to be functional, especially if the regulatory element is in a key cell type. And again, it's important to find these alleles so that we can focus our functional efforts on it. A kind of common strategy that one might use to build these enhancer gene maps is to look at regulatory elements that are themselves associated with gene expression. The approach that we use, which turned out to be very powerful, was to look at single-cell multimodal data, looking at data where we have both RNA data and ATAC data together, where we're getting a sense of how much activity we have in the regulatory element and how much activity we have in terms of gene expression. And one of the advantages of this is that it's very easily applied to disease tissues, because you can imagine you can go into an inflamed synovium and get all your cells out, get all the T cells out, and then build an enhancer gene map with this multimodal data. And this was work done by Saori Sakawi, who's now gone on to start her own lab at the University of Washington. And the way it works is we take single-cell multimodal data, as illustrated here. We build a map of it, and then we look for simple correlations between the gene and gene expression and ATAC data. We do this in a non-parametric way, and we do sort of single-cell modeling. You can imagine this sort of activity has been done quite a lot in the past, looking at bulk data, but that might be obscuring signals. So here you've got these blocks, which are supposed to be the bulk data, but it might be better if you have actually single-cell data and you model at the single-cell level. And we found in our hands that actually creates a lot more power. And so using this method, SENT, we see that scent really does a very good job at prioritizing causal variants in these various biobanks, both FinGen and UK Biobank, as well as EQTL studies, not shown here, compared to some of these other strategies one might use. And I found this really interesting. So this is the RBPJ locus for RA. And again, you see a whole bunch of SNPs, all in tight linkage with each other, hard to know which one is causal. But now when you build these scent enhancer gene maps, what you see is that in the T cells from RA, there's an enhancer that one of those SNPs overlaps with, suggesting that that's likely causal. Interestingly, if you look at T cells from PBMCs, you don't see that enhancer to be present at all, and you don't see it in other cell types, suggesting that when you build that enhancer gene map, it's not enough just to do it in T cells, but you have to do it in T cells, again, in the right functional context. And this is an interesting one, because if you look at that SNP and say, what's the downstream gene? It's not the closest gene right there. It's a gene that's a little bit further away that's involved in notch signaling, which is a mechanism that we've linked previously to RA biology. To summarize on this section, defining causal variation is the first step to getting a disease mechanism. I talked about multiple strategies. Statistical fine mapping might be useful to localize the causal variant, and this can focus functional efforts on a single genetic variant, and is particularly informative if the variant's a coding variant. In most instances, it isn't. Some variants co-localize with EQTLs, which doesn't define the causal variant necessarily, but it does give you a sense of what sort of molecular mechanism you should be looking for, and it helps you understand which variants are within enhancers and which ones they're linked to. Okay, so now if you have the causal variant, now maybe what you want to do is prove that it's causal. So in this next section, I'm going to present some really new work where we've been trying to functionally confirm the effect of causal alleles with genome editing. And this work was led by Yuri Baglienko, who's now faculty member at Cincinnati Children's Hospital, and Phoenix Mu, who's a postdoc with me in the lab. And what our aim here was really to validate non-coding allelic function. The idea is that we've been able to prioritize what we think is causal variation with all these computational tools like the ones I showed you in the previous section. But in general, we as a field don't really do a good job at trying to functionally confirm these causal variants. So there's a real need to start to do this, and we wanted to build a platform to do this. So like any reasonable person, we thought we would use like gene editing to get at this, and we started to think about sort of a typical strategy one might pursue. So you might start by obtaining what you think is a relevant cell type. Then you might define a genome editing strategy, some sort of CRISPR base editing strategy, for example. And then you might apply bulk RNA sequencing and look for differential expression compared to like non-targeted controls. And maybe that would tell you whether the allele is having an effect and what that effect is, and would help define the downstream genes. And there's some challenges that we came to appreciate as we pursued this. The first is that these genetic effects that we're looking for are relatively small, so your signal could get swamped out pretty easily. And one of the problems is when you're working with primary cells, as we often want to work with, the editing efficiency can be less than 100%, which means your signal is attenuated. And then finally, there's an issue of intrinsic versus extrinsic effects, which I'm gonna explain in these slides here. So when we're editing an allele, you might imagine we're splicing a T allele in here. We're very interested in the intrinsic effect, that you put the T allele in and the downstream gene expression goes up. Now, the reality is that a few cells may be edited. So if you've got your cells sitting in a pool of cells, you edit one of them, and what might be happening is that you might actually be perturbing all of the cells around you, because that cell is being disturbed, it's releasing cytokines or other substances, and that's creating a differential expression in the pool. So if the effect of the SNP is quite small and the editing efficiency is quite low, you might actually not see the effect that you want to see, but you might see other effects which are going to confuse you. So, to prove to you that this is a real concern, we look at this RPL8 EQTL locus. So this is a well-known EQTL. We focused on B-cell lines. There's a particular SNP that we think is the causal variant. It's not a known disease allele. It's just an EQTL locus. And what you can see here is that if you've got the C allele, you're going to have higher expression and the T allele lower expression. So just remember that, because we'll come back to that. So these are 3 different EQTL studies. And if you apply your fine mapping algorithm, you come up with the same SNP every time. So very likely that this one SNP is causal. So this is a great one to go after and do your editing experiment in. So we went ahead and got an AB8 editor put together. We altered the T alternative to the C reference allele, which would increase expression. And we looked at Dowdy cell lines, which are a common proxy. And we defined 5 pools. And we split the pools. So in one half of it, we did editing, and the other half, we did a non-targeted control. And then we did differential expression. We looked at RPL8, and we saw, there's no change, right? So, so that was a little disappointing. We thought, could it be the editing efficiency? That could be part of the problem, because we're only editing about 40% of the cells. But still, you would expect to see something. But now when you do differential expression, you see all kinds of genes are affected. So the gene that you think is the target gene doesn't seem to have an effect, and all of these other genes seem to be altered. And we wondered if we're looking at sort of extrinsic effects and not the actual effect of the SNP. So we want to do this in a different way. So we had a wish list. So we want to be able to introduce variants at base pair resolution. We want to be able to do it at scars, without scars. We want to directly read out the edited genomic loci. So we want to be able to sequence the DNA to see if we've made the changes that we think we need to make. We want to measure gene expression, of course, in the same samples. And we want to do this at the single cell level because we really want to be able to look at these really small effects of these EQTLs. So we came up with a strategy. Yuri named it Minecraft-seq, which stands for Multi-Omic Investigation of Nucleotide Editing with CRISPR by ADT flow cytometry and the transcriptome. So it's a little bit of a long name. The reviewers actually didn't like the name. They pointed out that there's some video game that people play, and we certainly wouldn't want to detract from the popularity of the video game. So we changed the name to CraftSeq. And it's a plate-based method where we're able to edit cells, and what we do is we get a genomic DNA amplicon, so we're actually able to sequence and get the genomic DNA. We're able to get gene expression data. We're also able to get cell surface marker protein, which is really important for immunological phenotypes, and we have index sorting, so we know exactly which cells have been CRISPR'd or non-targeted controls, et cetera. So what we're looking for is you might have a bunch of non-targeted controls and a bunch of cells that have been edited. You might see some differences. That's the red and the blue. But really what we're looking for is in the cells that you've tried to edit, some of them have been edited, some of them haven't. And what we're hoping to see is that as you go from AA to AG to GG, that you see a change in gene expression. Okay, so let's go back to the SNP. Let's go look at this RPL8 locus again. And now, you see that we've edited a bunch of cells. We're looking at a whopping like 900 cells here, so not that many cells. But you can see the vast majority of them aren't edited. They're in that reference TT. About a quarter of the cells have a single C allele added, and about 10% of the cells have 2 C alleles added. If you compare the control cells, the non-targeted control cells, to the CRISPR cells, you don't really see any difference in RPL8, exactly like the bulk experiment. But now if you look at those edited cells, and now we have the benefit, because we've sequenced the DNA, we know which ones have the edits in them. And now what you see is that as you add in C alleles, the expression of the gene goes up in a dramatic, statistically significant way. We model all of this with a single cell model using a negative binomial model. And now, because it's a genome-wide RNA-seq, we can actually test every single gene. And what you see is that the effect of the SNP is exactly on this one gene, RPL8, and it has no effect on any other gene in the genome, or at least no dramatic effect. And I'll remind you, this is a very well-powered experiment because every cell is basically its own experiment. So with 800 cells, we're able to see these signals. So we edited the RPL8 signal in Dowdy B cells with AB8. We observed low-efficiency editing. When we look at bulk differences in edit and control populations, we don't see what we want to see. But when we do single-cell modeling, we see that we're able to control for these extrinsic pool conditions, and we see a very specific effect on RPL8. So a second thing we thought we would try to think about is, can we look at 2 sets of variants at the same time. Can we look at two loci at the same time? So for this, we looked at this PAX5 gene. It's critical to B cell development. We again looked at Dowdy B cell lines. We examined two DNA binding regions of the PAX5 protein product. PAX5 is a transcription factor important for B cell development. So here we're looking, Vidya and Cassidy did this experiment. Here's PAX5. And what we did was we edited these two regions, one near the N-terminal region, one towards the C-terminal region, region B and region A. And we had a bunch of guides, so we introduced a bunch of random variants in both of these regions. And here what you see is that when we edited the cells, you can see that the CRISPR-celled and the non-targeted cells are kind of are sort of clustering together. And we have these sort of three clusters. And you see most of the non-targeted cells are all in cluster 0 with the gray bars. And most of the B-edited cells are in cluster 1 and cluster 2. And then we had some multiplexed edits where we had both edits in A and B together. So now we can go and look again at all the cells and see what sorts of variants we're seeing. So you can see we have 8 different variants, 4 in the A region, 4 in the B region. You can see some of the cells have multiple variants from A and B, again in that multiplex condition. And now we can do a simple single cell model where we model the effects of all of these variants together. And what you can see right away is that the B variants, particularly B25 and B29, are having pretty big effects on terms of gene expression and affecting a large number of genes, whereas the A alleles aren't having that much of an effect. And what you'll see is that there's a bunch of alleles that are grayed out. That's because they're synonymous alleles, so they have no effect on differential expression. And so this is one of the genes, J chain, which we know is a downstream target of PAX5. PAX5 normally suppresses it. As you introduce these alleles, you can see that the B29 edit in particular causes an increase in that expression of that gene. And you can see that there's a dosage-based component to it. And the other, the A edits and the synonymous edits don't have much of an effect. Because we're looking at multiple edits at the same time in the same cell, we can look for interactions. So we defined a score in terms of the effect of mutating this gene. We did it using the large number of genes that we see with B29. And what's really interesting is if you look at the cells with a B edit in it versus without the A edit in it, you do see a difference. So the effect of the A edit on the right, you can see is negligible when there's no B29 edit. But if you have a B29 edit, if you have an edit in the B locus, then the A locus starts to matter. So if the gene is already sort of broken up by an edit in the B region, then editing the A region starts to matter. So you're able to see these kind of interactions, and you can imagine this gives us the ability to look at synergistic effects all throughout the genome. So we present this CraftSeq multimodal assessment of RNA, DNA, and surface marker proteins. It's a powerful platform that really gives us the ability to define subtle effects of individual variants, the same kind of subtle effects that we see in genetic studies. And I gave some examples looking at a common regulatory allele, and I gave an example looking at multiplex variants within the same gene. And it's a plate-based method. It's a little bit laborious, but you're very well powered with a small number of cells. It costs about $3 a cell. And we're very focused on trying to expand throughput for this. But it has the real potential to examine disease alleles, particularly in different cell contexts, as I'll show in the next section. So in the final section, I'm going to talk about identifying SNP effects on molecular traits that are cell state specific. So we've been talking about EQTLs. Most autoimmune diseases, disease risk alleles, it turns out, aren't EQTLs. About 10 to 20%, probably closer to 20% of the non-coding autoimmune alleles are associated with the expression of a gene. The vast majority aren't co-localizing with an EQTL. And this is the problem of missing regulatory function as described by Shamil Sunyaev and others as we've looked at this. And for one of the possible explanations for this is that the effect of a SNP may be very context dependent. So a SNP is not necessarily affecting gene expression in most states, but maybe in a rare T cell state, it is having an effect. So it's possible that the effect of a SNP is state dependent. And for T cells, this is a particularly attractive idea since T cells take on different functional states. And disease conferring regulatory regions may be really only active within certain T cell states. This study led by Aparna was one where we looked at PBMC memory T cells from 250 individuals. It was a Peruvian cohort. We looked at about 500,000 memory T cell, 500,000 memory T cells. And we obtained RNA and surface marker protein, and we jointly modeled the RNA and protein using canonical components analysis. Using CCA, we found 7 important canonical variants. Normally, what we would do is cluster them together and define some, annotate T cell states. Here, we looked at the individual CVs and realized a lot of them did explain very specific T cell functions. And in our group in particular, we like thinking about T cells not as particular discrete states, but rather thinking about continuous states that capture their particular functions. So we're able to now look at these canonical variants and ask, do they interact with an EQTL effect? And here I just give an example of how we annotated these CVs looking at expression of specific genes. We can see that CV1 is capturing sort of cytotoxicity, CV2 is capturing regulatory T cells, and so on. And what we did now is we model EQTLs as a function of these CVs, and then we asked the question, do we see effects of SNPs on gene expression that are dependent on the T cell state? And so this is a particular EQTL. This is the MAF EQTL, and you can see that the effect of the SNP is explained by the color. And you can see that the strongest effect is on those red cells. And what you can do is you can actually grab the cells from each of these regions, and what you can see is on the left end, when you have the most naive, least cytotoxic T cells, the effect of the SNP is minimal. On the right, you can see the effect of the SNP on the expression of this gene is quite dramatic. When we looked at all of the EQTLs that we could find in T cells, about 1/3 of them were state dependent. So it was a surprisingly large number. And what's more striking is when you start looking at autoimmune disease alleles, the proportions are even larger. So about 70% of the RA EQTLs that co-localize with a T cell, RA disease alleles that co-localize with a T cell EQTL, 70% of them are dynamic or state dependent. And I really like this example because it shows you how this actually might matter. So this is a SNP near the CTLA4 gene. CTLA4, you can see on the right, is very highly expressed in regulatory T cells down at the bottom. It's those yellow cells on the right. But the EQTL effect is actually the biggest on the cells that don't express that much CTLA4. So this SNP actually results in like leaky CTLA4 expression. So it's not about, the SNP is not acting by increasing CTLA4 expression in regulatory T cells where we expect to see them. It's actually acting by increasing expression in the cells that normally don't express CTLA4. Can we experimentally validate a context-dependent T cell allele? So this is the IL-2RA locus. There's a SNP that we think is causal. It's associated with multiple autoimmune diseases. We looked at this SNP, and there's been conflicting literature about this SNP, whether it actually increases or decreases IL-2RA or the protein CD25. We hypothesized that it might be a context-dependent allele. So we used a base editor to introduce the SNP. We polarized the cells to Th1 and Tregs, and then we assayed 2,000 cells with Kraft-seq to see if we could see the effect of the SNP on gene expression and to see if the effect was specific to Th1s or Tregs. And indeed, what we saw is that the effect of the SNP is dramatic on both proliferating and non-proliferating Tregs, but not seen in Th1 T cells. And here we're looking at the CD25 surface marker, and you see sort of a parallel result if you look at the underlying gene, which encodes the CD25 protein. So two kind of consistent results, but Treg specific, not seen in Th Th1 T cells. So allele effects on gene regulation can be cell state specific. We demonstrate the power of single cell modeling to define these allelic effects, and about a third of EQTLs are dynamic within T cells. They're really enriched for autoimmune disease loci. And we also gave, I also showed you an example of how you can functionally validate this and look at state-specific effects using CraftSeq. That's the talk. So hopefully I've convinced you that a lot of these alleles that drive risk of disease are in fact acting in a state-dependent manner. So to conclude, defining the function of autoimmune disease loci is essential to really understand mechanism. And I've really focused on T cells, which is a central focus of our lab because of our interest in autoimmunity. We've focused on defining causal variation for immune-mediated disease alleles in T cells. It's essential really to focus on regulatory variation and functional confirmation of the effect of these non-coding alleles. I presented a protocol, CraftSeq, which is a single-cell multimodal strategy. And the really key thing is you have to sequence the genomic DNA and really know which cells got the edits that you want to introduce, because that's what really allows you to distinguish between intrinsic and extrinsic effects of editing. Alleles driving gene regulation are context-specific, and there may be many missed regulatory effects that are highly context-dependent that we're not seeing in our large-scale EQTL studies because the effect of the SNP is so specific for such specific cell types. And better therapeutics may be possible if we understand the context so that we can target a protein in a particular context, not just targeting it in a generic sense. I'll just acknowledge a few folks. So Yuri and Phoenix did a lot of the CraftSeq work. Yuri started that work, and Phoenix has kind of continued the project on. Aparna has done a lot of the dynamic EQTL modeling that I shared with you. And Jose has picked up that project and is trying to scale up our model so that we can apply it to larger and larger data sets. So as you guys build larger and larger data sets, we can keep up in terms of our single-cell EQTL studies. Sauri's done a lot of the enhancer gene construction. Alkis has been my longtime collaborator in statistical genetics. And David has helped us a lot with CRISPR editing, and Megan helped us put the T-cell cohort together. And finally, I'll also just acknowledge the whole lab, and I'll take some questions.
Wonderful talk. Thanks so much. I'm right here. But maybe open it up for a few questions. We have 5 minutes. Sean?
Amazing technologies for uncovering disease genetics, Shomo. A question about the conditional QTLs. I think you looked at a fairly broad T cell population. If you zoom in a little and try to look at conditional QTLs in just CD4 naive T cells or something like that, then does your number of conditional QTLs go down because your range of conditions is smaller?
Yeah, so, I think, so it's a little bit of a confusing question. So I would say, so for that study, we look specifically at memory T cells. So I think we find more condition-specific EQTLs in memory than naive T cells. I do think it's a little bit important how you make the choices that you make, right? Because you lose your condition-specific EQTLs for two reasons. Like one, you're losing heterogeneity, right? But the other thing is you're also losing your numbers as you start subsetting your cohort. Naive EQTLs, naive T cell EQTLs, I would imagine there would be many less because it's a fairly, famous last words, but I think it's probably a more homogeneous population.
All right, thank you.
Yes.
Very interesting presentation. The last point you made is kind of intriguing, that the allele effect on gene regulation can be cell state specific. We also are working on a similar situation, but the question is, do you see the chromatin accessibility change in the cell type where it is expressed versus which are not expressed?
So yeah, that's a great question. So, I think from what we've seen, again, we've not done a super careful analysis on this. Others have. I think what we generally see is that the ATAC QTLs are much less state dependent than the eQTLs themselves. I mean, obviously there's situations where the chromatin is closed and the gene isn't expressed. All of our modeling is looking for changes in sort of effect size. We control for differential expression. But we do sort of generally see that ATAC QTLs are kind of more stable within a cell type. A little bit hard, and one of the difficulties with those analyses, obviously the ATAC data is a lot more sparse than the RNA data, so there might be like a power issue.
Yeah, one last question, Wu Xiao.
Very, very nice talk. So I'm quite intrigued by CraftSeq. So people have been using, for example, MPR arrays in the past, but MPR arrays can only capture expression, enhancer, and promoter effect. And CraftSeq, in fact, can capture anything out of things, including that and outside of that. So I'm wondering, when you compare CraftSeq results versus MPRA results, what proportion of CraftSeq results can be replicated using MPRA results? And that really shows the power of CraftSeq, right?
Yeah, so, MPRAs we find, like at least in my experience, is pretty like non-specific to getting to causal variation. I mean, obviously MPRAs don't have the benefit of having chromatin context. I think Hilary Finucane has done some studies showing that MPRAs basically like increase the probability of a variant being causal by like twofold. So it does give you information, but it's sort of like more soft information. Here, obviously, you're looking at an allele in the context of the whole cell. Another thing I didn't talk about is with CraftSeq, because you're looking at a whole cell, you actually have the ability to pick up on trans effects. So if you edit an allele, you might see an effect on the cis gene, which is what I focused the discussion on. But if that gene has knock-on effects to other genes in the genome, you'll see that too. So I think that's another advantage of the editing approach.
All right, maybe just one last question from me, actually.
Okay.
In the context of the Human Cell Atlas, so you've shown beautiful work on genetics influencing gene expression. A lot of us here are doing just transcriptomics at the single cell spatial level. How important is it that we also perform whole genome sequencing, or is it sufficient to take SNPs from GWAS summary statistics, or should we also look into individual genome in the context of understanding genetic variation? And maybe if I may add, kind of what will be the impact of potential somatic mutations that one could acquire during their pathology?
Yeah, great questions. Yeah, I would say, getting whole genome sequencing, if you can do it, is a great idea. There's a lot of single cell data sets out there where there is no genotyping, right? I mean, we all know this. There's a lot of single cell data out there where it's just single cell. And if you want to do anything with SNPs, it's really difficult unless it's within the coding region. So I think at least getting genome-wide SNP typing is essential. Obviously, it doesn't give you a view of de novo mutations. Some of those de novo mutations have bigger effects. And I would say, in the context of how much it costs to do a single cell study, getting a whole genome sequencing done is like a drop in the bucket. So yeah, I would strongly advocate for whole genome sequencing.
Excellent, great. On that note, let's thank Soumya once again for a wonderful talk.